here's another example. We're just using a plant cell. What's one of the differences between a plant cell? So there's maybe two things in a plant cell will keep it from exploding, aside from pumps and stuff like that. We talked about one before. Vacuole, yeah, and the other one is the cell wall that will keep it somewhat rigid. So here we have a plant cell in a hypotonic solution. Hypotonic means it's starting to swell. In a hypertonic solution, it's starting to shrivel, it's trying to dilute whatever's outside of it. Okay, I'm just going to skip this slide, it doesn't really fit into the rest of what we did. Okay, who's this? Yosemite Sam. He's a big mustache, I find this guy looks hilarious. His name is Peter, how do you pronounce his last name? I think it's Hagen? I think it's Peter Hagen. And in 2003, he won a Nobel Prize in chemistry. So the thing that I've been talking about since the beginning, water can't just move through the membrane, it requires a channel. He's the one who discovered the channel, and he actually won a Nobel Prize for this. Does anyone want to describe the experiment that he did to discover this? No? I'll describe it for you. That's why I'm here. He used frog oocytes. And what he found, so after all of the experiments that we saw earlier on about cells going into hypertonic and hypotonic solutions, he found that if he took frog oocytes and he put them in any of these different solutions, so if you put them in a hypertonic solution, they weren't shriveling up. And if you put them in a hypotonic solution, they didn't begin to swell. So these oocytes are biological membranes just like any other. And he said, why? Why isn't it swelling like every other if water can just pass to and from the membrane? So eventually he discovered something called aquaporin. Okay, it's a particular gene that's found in most animal cells. Now, the experiment involved taking mRNA that coded for this protein called aquaporin. So everyone must be familiar with mRNA by now. So if you take DNA, mRNA is the step just before you turn your genetic information into a protein. This cell was able to take the mRNA from aquaporin and express it. In other words, it took the mRNA, it made a protein out of it, and this protein lodged itself into the biological membrane. So what happened? Can you see what happened? So what happens now? We have aquaporin. Of course you can't see that, so I didn't explain anything. This is, so you put it in a hypotonic solution, okay? The cells at the bottom do not have the mRNA of aquaporin, and the cells at the top do have the mRNA for aquaporin. So now can you tell me what happened? Yes? Yes?
It's part of mRNA from the above cell to the, like from above to below.
No, it didn't happen. It didn't transfer the mRNA.
The protein. After it.
No, that's not, it does look misleading. Maybe the other slide is more, can I go back? Yeah, if you were to separate them like this is just to show, the other slide was to show the control, but they weren't actually fusing, they weren't necessarily touching each other.
It's swelling up.
Yeah, the one expressing aquaporin is beginning to swell. Why?
Because water is rushing in.
Because water is rushing in. So in other words, what did we have to do for the water to flow into the cell? We had to put holes in it, we had to put channels in it, and that channel is called aquaporin. So this guy gets a Nobel Prize for that. You know how long this probably took to do? 10 minutes. I can do this in 10 minutes. I'm a doctor. It was easy. So this was in 2003. You'd be surprised a lot of the things that could change in just a few years. So I don't, it would probably be difficult to even do a thesis on this now. It's pretty simple to get mRNA to be expressed in certain cells. But I guess it's probably, I didn't read the paper, but it's, it changed the way we look at membranes, right? You saw that slide earlier on. They thought that water was just flowing. Peter's as well, but not that I do that. So, that's aquaporin. So you guys will have to recognize the structures in aquaporin. This is a top view. Okay, an aquaporin molecule is usually made of four different, it's made of four monomers. Okay, and this is water passing through the different channels. All right. So this is present in most animal cells. Now the question here is, considering what we've discussed above, one of the things that I mentioned right at the beginning of the class, why would water, how do these channels work to select for water, to allow water to pass through? Does anyone have any ideas? Pardon? Something like that. So I guess the more general answer would be this channel, okay, these channels create an environment that's more favorable to water. All right? So normally the hydrophobic lipid tails inside the membrane are not favorable to the passage of water. So these channels are making it favorable to the passage of water. How do they do that? It's pretty interesting. We're going to look at these, at this diagram from the Nobel Foundation. What they found is that, so there's a lot of conserved arginine and histidine residues. So you guys have taken biochemistry, maybe you have an idea of what the shape is of these amino acids. But particularly what you want to look at is the shape and structure of asparagine. And what they found is that the asparagine residues in this channel were forming hydrogen bonds with the water. Okay? So this actually creates a very favorable environment. It means that when the water is moving through there, it's like, I'm comfortable here. I can just form hydrogen bonds and stick around, and we can move to the next asparagine residue and the next one and the next one down my concentration gradient. In other words, thermodynamically, if it's more favorable for water to move from there to there, this is providing an adequate environment of travel. Does everyone understand that? An analogy would be, I don't know, I don't think you need an analogy. If you're passing through, you know, you would, never mind. So I think that would be general, and keep this in mind when you're looking at any of the channels. The idea is to create a favorable environment for the passage of whatever molecule we're looking at, whether that be water or a particular ion. So you guys are going to see this slide probably when you go online. Don't be too scared of it. It's just showing you here, you know, this is a water molecule, and it's forming hydrogen bonds with these asparagine residues. And what would happen is, as the water moves here and forms a hydrogen bond, you know, as water continues to move down the channel, it will eventually form hydrogen bonds on the residue lower, lower, and lower until it passes all the way through the channel. Okay? Okay, there are different types of gated channels. I don't know if you guys looked at this in cell biology. You can have a voltage-gated channel. In other words, it opens or closes in response to the electrochemical potential across a membrane, and we'll look at electrochemical potential in a second. There's a ligand-gated channel. You guys will see. Did you guys look at action potentials? No? Okay, I'm sure you'll look at it later. It's a fun lecture. You did? Someone says you did. In fact, you probably saw it in cell biology. But anyway, action potentials are when something binds to a ligand to open up a channel, then that causes a change in the electrochemical potential across the membrane and causes other channels to open. And that's how you move. That's how your nervous system works, by action potential. Okay, that's not part of this lecture, but you will probably need to understand this to get that later on. So ligand-gated, it means that some molecule is binding to the channel to cause it to open. And you can see that here. Binds, opens. Ligand-gated intracellular, whatever, it's just saying that now the ligand is coming from inside of the cell instead of outside of the cell. Okay? And mechanically gated, in other words, you know, whatever, if I were to flick someone, the pressure from that is going to cause a change in conformation in sensory proteins, and that might be an action potential as well. Okay? So what we're going to look at in some detail are ion channels. So the direction of transport in ion channels is generally determined by the electrochemical gradient. So I'm going to look at that now. I think I'm going to use the blackboard